Good afternoon one and all. So yes, so far from this chapter cells and tissues, we have dealt with the introduction of the cell theory and completely about prokaryotic cell, and we have also dealt with the differences between prokaryotic cell and the eukaryotic cell. So now we are going to begin with the eukaryotic cell, where first we will clearly understand the cells of both plant cell and animal cell and the cell organelle distribution in both plant cell and animal cell, how the compartmentalization of cytoplasm is given, what is endomembrane system, this and all we will be learning first, looking into plant cell and animal cell. So first let us clearly understand the plant cell. So plant cell, if you have to see plant cell whose outer wall is composed of cell wall. The limiting boundary of a plant cell is what? Cell wall. Next to the cell wall we have what? Cell membrane. So the outer limiting boundary of a plant cell is cell wall. Next to that we have what? Cell membrane. So inside the cell membrane, this complete fluid filled matrix is called as cytoplasm. Cytoplasm is the fluid filled matrix. Fluid filled matrix. So this fluid filled matrix cytoplasm has got the compartmentalization, clear compartmentalization of cytoplasm is observed, wherein at the center or inside the nucleus, inside the cytoplasm, the major organelle is nucleus. Nucleus, which is double membrane bound organelles, is the hub for the storage of genetic information. Nucleus organelle will have four major parts inside it. Those are nuclear pore, nuclear envelope first, nuclear envelope followed by nuclear pore, and then nucleoplasm, fine, and nucleolus. These are the components of a nucleus. A nucleus has these four components: nuclear envelope, nuclear pores, nucleoplasm and nucleolus. The extensions of the nucleus are, the extensions of the nuclear envelope is endoplasmic reticulum. Where is see, rough endoplasmic reticulum and smooth endoplasmic reticulum we have as such. So these are the extensions of the nuclear envelope. So these are the network of reticular structures which are extending from the nuclear envelope. So this network of reticular structures, if they have ribosomes over it, then it is called as rough endoplasmic reticulum. It has ribosomes over it. This is purely involved in protein synthesis. And then smooth endoplasmic reticulum is involved in lipid synthesis. This is about the nucleus and extensions of nuclear envelope, both rough endoplasmic reticulum and smooth endoplasmic reticulum. And then if you see still the single membrane bound organelles inside the cytoplasm of plant cell, we can observe Golgi bodies, which are the sites for the modification of lipids and proteins that are being synthesized from RER and SER. After Golgi bodies, it is the organelle vacuole and lysosomes. These are the single membrane bound organelles. Vacuole in a plant cell will occupy 90% of the plant cell's volume. Okay, 90% of the plant cell's volume is occupied by what? Vacuole. Fine. And then still single membrane bound organelles, what you can see? Peroxisomes, microbodies, these are present inside the plant cell. There is no centriole. Okay. And what are the amembranous organelles or non-membrane bound organelles? Non-membrane bound organelles include only ribosomes. There is no centriole inside the plant cell. Fine ma? And then what are the double membrane bound organelles that you can see? Nucleus, chloroplast and mitochondria. These are the double membrane bound organelles that are observed in a plant cell. So, this is the clear structural description, and here you can see some connections called as plasmodesmatal connections. These plasmodesmatal connections will traverse across the cell wall and connect the cytoplasms of two different cells. Okay? They will connect the cytoplasms of two different cells. So, here once let us make a list of non-membrane bound, single membrane bound and double membrane bound organelles in a plant cell. Okay? Plant cell, non-membrane bound, double membrane bound and single membrane bound organelles. So, here if you have to see first amembranous or non-membrane bound organelle. Bound organelles. Single membrane bound organelle, second. Single membrane bound organelles. And third includes double membrane bound organelles. What are the non-membrane bound organelles that are present inside the plant cell? The only non-membrane bound organelles that are present inside the plant cell is ribosomes. What type of ribosomes you can observe? Ribosomes are of two types inside the plant cell. We have 70S ribosomes and 80S ribosomes. 80S ribosomes are present in the cytoplasm of a plant cell and above rough endoplasmic reticulum. In these sites 80S ribosomes are present, wherein larger subunit is 60S and smaller subunit will be 40S. And then 70S ribosomes, if you have to see, the larger subunit of 70S ribosomes is 50S and smaller subunit is 30S. Fine. And these ribosomes are present inside, these ribosomes are present inside chloroplast, stroma and mitochondrial matrix, mitochondria. In these two organelles 70S ribosomes are present. This is the complete description about the ribosomes which are present in the plant cell. These are the only non-membrane bound organelles that are being observed in the cytoplasm of a plant cell and in chloroplast and mitochondria. Here if you see single membrane bound organelles. What are the single membrane bound organelles that we have come across? First is endoplasmic reticulum, both rough endoplasmic reticulum and smooth endoplasmic reticulum. These are the first thing. Second is Golgi bodies. Third will be lysosomes. Fourth is vacuoles, lysosomes, vacuoles. These are the single membrane bound organelles. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi bodies, lysosomes, vacuoles and peroxisomes. Fine. These are the single membrane bound organelles that are being observed in a plant cell, of which rough endoplasmic, smooth endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles will form an endomembrane system. Okay, endomembrane system. About endomembrane system we will discuss in detail in our coming class. Okay, of which single membrane bound organelles, those are vacuoles, will occupy 90% of the plant cell's volume. Fine. Now, double membrane bound organelles. Double membrane bound organelles include chloroplast, mitochondria, and nucleus. Chloroplast, mitochondria and nucleus. These are the double membrane bound organelles that are being observed in a plant cell. So, this is complete organelle distribution inside a plant cell, non-membrane, single membrane and double membrane bound organelles. And we know the outermost limiting boundary of a plant cell over cell membrane is cell wall. Next to that, cell membrane or plasma membrane is present, and traversing the cell walls we have plasmodesmata, which are the cytoplasmic connections in between the cytoplasm of two plant cells. Next is the animal cell. Let us look into the clear structural description of an animal cell. So, animal cell, if you see, the outermost limiting boundary of an animal cell is cell membrane. Cell membrane is the outermost limiting boundary, and this diagram is the cell of your small intestine where it is showing the microvilli. So, the outermost limiting boundary is what? Plasma membrane, and the double membrane bound organelles that are being observed inside an animal cell includes nucleus. Inside nucleus, 4 components we have discussed: nucleoplasm, nucleolus, and surrounding that you have nuclear envelope, and in which you have nuclear pores. And then one more organelle that is double membrane bound, that is mitochondria, which is double membrane bound organelle called as powerhouse of the cell. And then single membrane bound organelles include smooth endoplasmic reticulum, rough endoplasmic reticulum, and Golgi bodies or Golgi apparatus, lysosomes, and vacuoles. Vacuoles are present in very small and negligible amounts, not like that as you see in an animal cell, where there it is occupying 90% of total cell's volume. And what are the non-membrane bound organelles that are observed in an animal cell? Centrioles are the non-membrane bound organelles. Centrioles are absent in a plant cell, and ribosomes are the non-membrane bound organelles that are being observed. So, now let us also look into the clear description of organelles, distribution of organelles inside an animal cell. Animal cell has non-membrane bound, single membrane bound, and double membrane bound organelles. Single membrane bound, and double membrane bound. First, look into non-membrane bound organelles. As we have discussed, the non-membrane bound organelles, centrioles. Centrioles are nothing but these are two perpendicular cylindrical structures which are present inside the centrosome. Centrosome does not have any membrane, it just has pericentriolar space. Inside this you have two cylindrical structures which are perpendicular to each other with 9+0 microtubular arrangement. The major function of this centrioles include, formation of spindle apparatus or spindle fibers. And second non-membrane bound organelles include ribosomes. And as we have discussed, what type of ribosomes are present? Both 70S and 80S type of ribosomes are present. Okay. 70S type of ribosomes include, 70S type of ribosomes are present in mitochondria. 80S type of ribosomes you observe above the rough endoplasmic reticulum and inside the cytoplasm. And what will be the larger subunit of 80S ribosome? 60S, and smaller subunit will be 40S. Larger subunit over here is 50S and smaller subunit is 30S. These are the non-membrane bound organelles that we see in an animal cell. And here if you see single membrane bound organelles. Single membrane bound organelles include of course primarily it is rough endoplasmic reticulum, smooth endoplasmic reticulum, and then Golgi bodies or Golgi apparatus, then lysosomes, and then vacuoles, peroxisomes, and microbodies. These are the single membrane bound organelles that are observed in an animal cell. Next, double membrane bound organelles include nucleus and mitochondria. This is the complete organelle distribution that is observed in an animal cell. So, now it is clear for us both the organelle distribution of a plant cell and an animal cell. So, clear compartmentalization of cytoplasm is clear to us. Now, let us look into the differences between a plant cell and animal cell. The primary difference is cell wall, okay, cell wall, which is also said in the cell theory by Schwann, okay, wherein cell wall is the limiting boundary of a plant cell, and mostly plant cell wall is made up of cellulose, hemicellulose and then pectin. Cell wall is absent inside an animal cell. Then shape of the cell is also different. In animal cells, the cells are irregular and rounded, whereas plant cells generally fixed, rectangular or polygonal. Plasma membrane, if you see, plasma membrane is present in the plant cell, which is inner to the cell wall. Plasma membrane is present as the outer boundary of an animal cell or limiting boundary. Plastids include, plastids are present, for example, chloroplast in green plants, or it can be chromoplasts or leucoplasts as well. And then in animal cells, plastids are absent. Vacuoles, they are large central vacuole, single large central vacuole is observed in the plant cell, which occupies 90% of total cell volume, and this acts as a dump yard inside the plant cell where all the excretory products are being dumped into the vacuole. That is why it is also called as sap vacuole. The membrane which is surrounding this central vacuole is tonoplast. Okay, it is a single membrane bound organelle, and the membrane which is surrounding this is tonoplast. Next, here in animal cells, the vacuoles are small and temporary. Next, centrosomes and centrioles, they are absent in a plant cell and present in a animal cell. So, in animal cell, this aid in the function of what ma? Spindle apparatus and spindle fiber formation. The spindle apparatus or spindle fibers will help in the cell division of an animal cell. Then how cell division process is mediated in a plant cell in the absence of the centrioles. Plant cell will be having microtubular organizing complex. Microtubular organizing complex. So, this microtubular organizing complex will aid in the process of cell division in plant cells. Plant cells, though in the absence of centrioles, it will conduct the cell division with the help of microtubular organizing complex. Microtubular organizing complex. Clear till here, right? Ma? Next. Lysosomes, which are rarely present or sometimes animals absent in the plant cells, which are actively present in the animal cells. And in animal cells, we can come across primary lysosomes, secondary or heterophagosomes, residual bodies and tertiary lysosomes as well. And in Golgi bodies or Golgi apparatus, you see, in plant cells, you can see the Golgi bodies like this, which are not interconnected. Okay, the Golgi bodies are not interconnected as that you see in a plant, as that you see in an animal cell. So, these cisternae which are not interconnected inside the plant cell, which are present as distinct units, these are called as dictyosomes. Okay, dictyosomes are present in the plant cell. Dictyosomes are nothing but Golgi bodies which are not interconnected. Next, in animal cell, Golgi bodies are present as a single well defined unit. Okay, nucleus position in plant cell is peripheral, it is pushed up because of vacuoles, and in animal cell it is at the center. Energy stored in the form of starch, here energy stored in animal cell in the form of glycogen. Cytokinesis is through cell plate formation, and this is through cell furrow formation. How you see, in an animal cell at the end of telophase, a small furrow is developed from the periphery. This furrow deepens to the center. Okay, as such, two cells will be divided. Karyokinesis and cytokinesis happens. So, this pattern of division from peripheral end to center through cleavage furrow or the furrow formation is centripetal mode of cell division, okay, centripetal mode of cytokinesis. But then in plant cell, if you see, in plant cell, in between two cells, at the center first cell plate is formed. This cell plate from the center it will move towards the peripheral ends. Cell plate from the center it will move towards the peripheral ends, and this gets matured to middle lamella. Middle lamella is the first formed layer in between the plant cells. Of course, we will look into how the cell wall formation also, but here as of now you remember, first cell plate is formed at the center. This moves from the center towards the periphery, and that is how a cell plate is formed first. Later it matures to middle lamella. This mode of cell wall formation or cell division is called as centrifugal. This is called as centrifugal mode of cell division. And then chloroplast. Chloroplasts are present in the photosynthetic cells, absent in an animal cell. Tonoplast is a membrane bound vacuole. Here of course vacuoles are negligible, right. Next, last today we are going to discuss is cell wall, fine. So, cell wall is observed in all the types of cells, okay, we have discussed also. Cell wall is the major criteria under two kingdom system of classification. So, all those organisms which have cell wall are placed under one kingdom, that is Plantae by Carolus Linnaeus in his two kingdom system of classification. So, what all those organisms which has cell wall? It includes bacteria. Bacteria, though it is a prokaryotic cell, it has cell wall. Next, fungi. Fungi has cell wall. Algae has cell wall. Higher plants also have cell wall. So, all these were placed under the kingdom Plantae under two kingdom system of classification. Now, let us recall the composition of cell wall. Bacterial cell wall composition is peptidoglycan. Fungal cell wall composition is chitin, which is also called as fungal cellulose. It has NAG units, N-acetyl glucosamine units. If you take unicellular fungi like yeast or Saccharomyces cerevisiae, its cell wall is composed of cellulose only. Fine. Algae. Algal cell wall is made up of course, in green algae though it has outer pectose layer and inner cellulose layer. Brown algae will have cell wall is composed of cellulose, but then its outer wall above the cellulosic cell wall it has hydrocolloids like algin. And red algae, if you see red algae, it also has cellulosic cell wall, but above the cellulosic cell wall it also has hydrocolloid composition like agar, carrageen and fumarate. Agar, carrageen and fumarate. And then higher plants cell wall composition includes cellulose, hemicellulose and pectin. Fine. The cell wall composition is important for us. And look at here, as I told you, the cell wall formation or cell plate formation is from the center towards the peripheral ends like this. This is centrifugal mode of cell division. Okay. First layer that is formed is what? Middle lamellae. Middle lamellae is the first layer that is formed inside the plant cell, in between cells. So, here can you see the middle lamellae clearly? This is the middle lamella layer that is formed inside the plant cell. Next, inner to the middle lamella, primary cell wall is formed. Inner to the middle lamella, primary cell wall is formed. Next, inner to the primary cell wall, secondary cell wall is formed, that is close to the cytoplasm. Okay, I will explain you clearly. See, first in between two cells, first layer that is formed is what? Middle lamella. Consider this as cytoplasm. Okay, inner to the middle lamella, the layer that is formed is primary cell wall. Okay, this primary cell wall is elastic, thin and extensible, and inner to the primary cell wall, secondary cell wall is formed. Okay, inner to the primary cell wall, secondary cell wall layers are formed. Okay, secondary cell wall layers are 2 to 3 layered secondary cell wall. So, as the secondary cell wall grows, this primary cell wall will lose its structural integrity. Okay, so this is how the wall layer formation takes place. So, after the formation of secondary cell wall, if you have to see cell walls from the cytoplasm to periphery or from the periphery to cytoplasm, look, from the cytoplasm to outer side, if you have to see close to the cytoplasm, which cell wall layer is present? Secondary cell wall. Okay. Next to the secondary cell wall you have primary cell wall. Next to the primary cell wall you have middle lamellae. Fine. From out to in, if you see, outside it is what? Middle lamellae. Inner to the middle lamellae you have primary cell wall, inner to the primary cell wall, secondary cell wall, and secondary cell wall is close to cytoplasm. This order should be clear in our mind. Okay. So, with this, cell wall is clear to us. So, that's all for today's session. So, in today's session what did we learn? We have looked into the clear structural composition of the cell in a plant cell and clear organelle distribution in a plant cell. After that, we have looked into animal cell, composition of organelles, then non-membrane bound, single membrane bound and double membrane bound organelles in an animal cell. After that, we have clearly looked into the differences between plant cell and animal cell, and then about cell wall. What are the different organisms which have cell wall, in which they are placed into kingdom Plantae under two kingdom system of classification by Carolus Linnaeus, their cell wall composition, and how the cell wall layers are arranged inside a plant cell. Okay. So, with this we will end up today's session, and in tomorrow's session we will look clearly about cell membrane and endomembrane system. Okay. That is all for today. Thank you.